Hello everyone, welcome to Biotech Notebook. In this video, we are going to see in detail about a special enzyme restriction endonucleases. Okay, so restriction endonuclease, also known as restriction enzyme, it is a protein which is naturally produced by bacteria. Thousands of restriction enzymes have been identified and isolated from various bacteria. In this, hundreds of restrictions enzymes are commercially available for laboratory use. The function of this restriction endonuclease is it cuts double-stranded DNA into smaller fragments at or near specific nucleotide sequence. This specific nucleotide or DNA sequence is known as restriction site. Okay, restriction sites are nothing but a specific DNA sequence where the restriction endonuclease make a cut. Okay, example, the restriction endonuclease enzyme ECOR1, which is isolated from the bacterium E. coli, which recognizes the following sequence: 5' GAATTC 3' opposite strand 3' CTTAAG 5'. Each restriction endonuclease is very specific to recognize the sequence, and also it make a specific cut. This ECOR1 cut in between G and A on both the strand. Okay, thereby it make it into two smaller fragments. Then these nucleotide sequence recognized by these restriction endonucleases are palindromic sequences. Palindromic sequence means a sequence of nucleotides. Okay, a sequence of nucleotides that reads the same forward and backwards. Okay, if you read in forward direction, it is GAATTC. Okay, same if you read it in backward direction. See, same GAATTC. Okay, or otherwise you can tell as both strands read same when moving from 5' to 3' direction. So 5' to 3' direction it is GAATTC. In this 5' to 3' direction also it is GAATTC. So this type of sequence is known as palindromic sequence. Okay, then this restriction endonuclease enzyme is also known as molecular scissor because it cuts DNA like a scissor into smaller fragments at a specific nucleotide sequence. Next, let's say how restriction enzyme cut DNA at its recognition site. Okay, so there are three steps involved in this process. The first one is recognition, second one is binding, then third one is cleavage process. Okay, so in recognition step, the restriction enzyme scans the whole DNA for its recognition or restriction site. Okay, that is your restriction enzyme scans for a specific palindromic nucleotide sequence. After recognition of your restriction site, the restriction enzyme goes and binds to the double-stranded DNA at the restriction site. Okay, then it cut both the strands of the double-stranded DNA at or near the restriction site based upon the type of restriction enzyme, and it creates sticky end or blunt end. Okay, there are two types of ends made by this restriction endonuclease enzyme. So next, let's see the difference between these sticky end and blunt end. Example for sticky end making restriction enzyme is ECOR1, which is isolated from E. coli. And example for blunt end making restriction enzyme is H3, which was isolated from Haemophilus aegyptius. Then this ECOR1 recognizes the restriction site in double-stranded DNA sequence is GAATTC from 5' to 3'N. This H3 recognizes the sequence in double-stranded DNA is GGCC from 5' to 3'N. Then its cleavage site. Okay, so restriction site is the nucleotide sequence which is recognized by the restriction enzyme is known as restriction site. Okay, whereas cleavage site means the location where it make a cut in a double-stranded DNA is known as cleavage site. Okay, so the cleavage site of enzyme ECOR1 is in between G and A on both the strand at the 5' end. Then cleavage site of H3 enzyme is in between G and C. ECOR1 enzyme cleaves in between G and A on both strands and it produces these two fragments. And H3 enzyme cut in between G and C and it makes these two fragments. ECOR1 makes an uneven cut, whereas your H3 makes a straight cut. Okay, so this type of cut is known as staggered cut, that is uneven cut, and this is known as straight cut. Okay, then this uneven cut makes these two fragments. If you look at these two fragments, you may notice there is a 5' overhangs of 4 nucleotides at 5' end. Okay, this is your fragments made by these echo Orban enzyme. So here you may notice that it has 5' overhangs of 4 nucleotides. Okay, so this overhangs means a single stranded region. Okay, so TTAA, the last 4 nucleotides or single stranded region of DNA at the end of double stranded DNA molecule. Okay, so both the fragments have 5' overhangs of 4 nucleotides. Okay, AATT. Okay, so overhangs means at the end of your double stranded DNA molecule you have single stranded nucleotides. These nucleotides at 5' end are known as 5' overhangs. Okay, so these single stranded overhangs are complementary to each other. Okay, so this 5' to 3' it is AATT, whereas 3' to 5' it is TTAA. So these single stranded nucleotides are complementary to each other, so they are said to be cohesive or sticky ends. Why? Because you can ligate these two fragments. You can join these two fragments by using DNA ligase enzyme. So if you cut a DNA with sticky end making restriction endonuclease, you can efficiently join those two fragments by using an enzyme DNA ligase. DNA ligases make hydrogen bonds between these complementary bases. Okay, so restriction enzyme act as molecular scissor to cut a DNA at specific sequences, while DNA ligase is the molecular glue that joins the DNA fragments back together. So these two enzymes together essential for genetic engineering. Okay, scientists uses the same restriction enzyme to cut a gene and a plasmid. They will insert a gene into the same plasmid to create a recombinant DNA molecule. Specifically, if you cut with sticky end forming restriction endonuclease, both will create 5' overhangs. So it will be easy to insert this gene of interest into the plasmid by using DNA ligase enzyme. Okay, so thereby scientists can create recombinant DNA molecule. Whereas in blunt and your cut is straight, so here there is no overhangs. So joining these two fragments with DNA ligase is less efficient. Then the method of this cleavage process is hydrolysis. Here the restriction enzyme catalyzes a hydrolysis reaction. Hydrolysis reaction means it uses water molecule to break the phosphodiester bond present in the nucleotide sequence. This is your hydrolysis reaction. Restriction endonucleases catalyzes this hydrolysis reaction. So restriction endonuclease won't directly involve the reaction, it just catalyzes this hydrolysis reaction. This hydrolysis reaction uses water molecule to break this phosphodiester bond, thereby it produces 5' phosphate group on one side and 3' hydroxyl group of the other. Okay, this phosphodiester bond is the linkage between the 5' phosphate group of one nucleotide and 3' hydroxyl group of another nucleotide. Then next let's see what is the main function of this restriction enzyme in bacteria. We already seen that we are isolating this restriction enzyme from bacteria. So what is the function of this restriction enzyme in bacteria? That is, in bacteria this restriction enzyme serves as a defense mechanism against bacteriophage. Okay, so in bacteria it produces this enzyme to kill foreign invaders, bacteriophages. Okay, so bacteriophages are viruses that infect bacteria. So when bacteriophage DNA enters a bacterial cell, okay, so once bacteriophage infect the bacteria, it starts to release its DNA into the bacterial cell. Okay, for example, if you consider E. coli, then the restriction enzyme in E. coli is echo Orban. Okay, so once bacteriophage DNA enters into a bacterial cell, the restriction enzyme echo Orban recognizes the foreign DNA and it searches for its recognition site in foreign DNA. Okay, once it recognizes its restriction site in foreign DNA, it make a cut in restriction site, then it forms fragments of DNA. Thereby it prevents viral replication. Then host DNA protection. That means the echo Orban's restriction site is not only present in this foreign DNA, it also present in bacteria's own DNA. Okay, so the bacteria's own DNA is protected from this echo R1 by the process DNA methylation at the recognition site. Okay, so this methylation process prevents the restriction enzyme from recognizing and cleaving the restriction site present in bacteria's own DNA. Okay, so in DNA methylation process, methyl transferase, okay, a special enzyme add methyl group to the restriction site present in the own DNA. Okay, thereby it prevents the restriction enzyme from recognizing and cleaving this restriction site. So this is restriction modification system in bacteria. Restriction enzyme present in bacteria used to recognize its restriction site in foreign DNA. Okay, so this restriction site is in bacteriophage and it cuts in between G and A and it make bacteriophage DNA into smaller fragments. Okay, whereas modification system recognizes the same restriction site, echo R1's restriction site in bacteria's own DNA. Okay, so this is the sequence present in bacteria's own DNA. This is the sequence present in bacteriophage DNA. Okay, so the sequence in bacteriophage DNA is cut by echo R1 enzyme, whereas the echo R1 restriction site present in bacteria's own DNA is protected by echo R1 modification enzyme, that is methyl transferase, by adding methyl group to adenine or cytosine base. Okay. Next, let's see types of restriction enzymes. You have 4 types of restriction endonucleases: type 1, type 2, type 3 and type 4. This type 1, type 2, type 3 cut DNA at or near specific DNA sequences, but type 4 in restriction endonucleases cleaves modified DNA such as methylated, hydroxymethylated and glycosylated methylated DNA. Okay, type 4 restriction enzyme cleaves only methylated DNA or hydroxymethylated DNA or glucosylated DNA sequence. Okay, let's see the difference between type 1, type 2 and type 3. Okay, type 1 restriction enzymes are large and multi-subunit complexes. Type 3 also large and multi-subunit complexes. Okay, type 1 and type 3 enzymes share some common features. Type 2 restriction endonucleases are simple in nature. Then type 1 has multifunctional, that is both endonuclease, that is it cut the DNA, and also it has methylase activity. The same enzyme itself can do both endonuclease and methylase activity. Methylase activity is it will add methyl group to its own DNA. Okay, but type 2 restriction enzyme has only endonuclease activity. It cut at the specific recognition site. Type 3 also has both endonuclease and methylase activity. Then type 1 cleaves DNA at random site. Okay, it won't cut at a specific site, so it doesn't have more application. Type 2 cleaves at the restriction. So type 2 restrictions enzymes are more specific in their cutting specific DNA sequences. These type 2 restriction enzymes have wide application in genetic engineering field. Type 3 restriction enzyme cut 25 base pair away from the recognition sequence. Okay, so once it recognizes the sequence, from that sequence 25 base pair away it will make a cut. Then this type 1 restriction enzyme requires the energy of ATP for cleaving the DNA. But this type 2 restriction enzyme requires no energy of ATP. It requires only Mg2 plus for its cleavage process. Type 3 restriction enzyme also requires energy of ATP. Then example for type 1 restriction enzyme is echo B, and it recognizes the sequence from the 5'AAC, and in between they have random 6 nucleotides. 3'N it has GTGC. So this is the recognition sequence of ECOB. Then echo R1's recognition sequences from 5' to 3' it is GAA-TTC. Then type 3 restriction endonuclease echo P151. Recognition sequences CAG, and it will cut 25 base pair away from this recognition sequence. Compared to type 1, type 3 and type 4, type 2 restriction enzymes are more specific. Type 2 restriction enzymes make a cut at a specific recognition site. So it has more applications in gene cloning, DNA analysis, DNA sequencing, RFLP, that is restriction fragment length polymorphism, and DNA mapping. Let's see in detail about these type 2 restriction enzyme. Thousands of type 2 restriction endonucleases have been discovered in different bacterial species. More than 100 different DNA sequences are recognized by one or more of the enzymes. Recognition sequence of these type 2 restriction endonucleases are usually 4 to 8 base pair long, and it is palindromic. Okay, so palindromic means it read both forward and backward the same sequence. Example, hint 3 read from 5' to 3' direction AAGGCTT. Okay, AAGCTT. Then we can further classify type 2 restriction endonuclease into 3 types: isosomers, neosomers, isocadmers. So it is based on how they recognize and cleave the DNA sequences. Okay, isosomers or pairs of restriction enzymes that recognize the same recognition sequence, same restriction sites, and cut it at the same position. Okay, so isosomers or pair of restriction enzyme, example SPE1 and BCU1 or isosomers. These two restriction endonucleases recognize same recognition site or restriction site, and it leaves at the same position between A and C from 5' end. Then it both produce sticky end. The next one is neosomers. Neosomers or pairs of restriction enzymes, example SMA1, XMA1. They recognize the same DNA sequences. Okay, both recognizes the same DNA sequence, GGG, CCC. Okay, GGG, CCC. But they cut at different positions. Okay, so SMA1 cut in between G and C from 5' to 3' direction, but XMA1 cut in between G and G. So here SMA1 will make a straight cut, so it produces blunt end, and XMA1 will make an uneven cut, that is staggered cut, so it make a sticky end, cohesive end. End is known as non-cohesive end and sticky end is known as cohesive end. Okay, because sticky end, after cleavage you can stick easily again it with DNA ligase enzyme, but blunt end it is difficult to ligate after cleavage. Blunt ends are also known as non-cohesive ends. Sticky ends are known as cohesive ends. Then isocadmers, these are pairs of restriction enzymes that recognize a different DNA sequence but produce the same ends. Okay, for example, SAU3A recognize GATC. It make a cut upstream of your base G. Okay, at 5' end. And BAMH1 recognize GGATCC. Okay, so here common bases are the 4 bases GATC. Here also it has the common same base GATC. Okay, so BAMH1 make a cut in between GG, so it make GATC overhang. Here also you have GATC overhang. So these SAU3A and BAMHI both produce sticky ends. Next, let's say naming of restriction enzyme. So the first letter of the name is the first letter of the bacterium genus. Okay, for example, Echo-Orban, which is isolated from the bacterium Escherichia coli. So the first letter of this enzyme denotes the bacterium's genus. Okay, so first letter of genus. Next two letters or the first two letters of the species name. Okay, so here species name is coli. Okay, genus name is Escherichia, species name is coli. Okay, so the first letter is first letter of genus, and the next two letter is the first two letter of species. So if you consider the another example, hint 3, which is isolated from Haemophilus influenza. So the first letter is the first letter of genus Haemophilus, and the next two letter IN is the first two letter of the species influenza. Then the subsequent letter or number indicates the specific strains of the bacterium. Okay, here the specific strain of the bacterium is R. So the third letter is specific strain of the bacterium. Okay, so here R comes from the strain or Y13. Okay, in hint 3, D is the strain serotype D. Then the last one is a Roman numeral at the end, shows the order in which the enzyme was isolated from this particular strain. So 1 means first, 2 means second, okay, and so on. So in Echo-Orban, 1 means first enzyme isolated from this strain. Okay, in hint 3, 3 means this is the third enzyme isolated from this strain. Some list of sequences recognized by type 2 restriction enzymes. Okay, so BAMHI, which is isolated from Bacillus ameloquaciens, and this is the sequence recognized by this BAMHI, and it cuts in between G and it makes sticky ends. Okay, then next one is CLA1. Okay, so CLA1 is isolated from cryophanon lactam L, and this is its recognition sequence. It cuts between T and C. It also makes sticky end. Then echo or V is an example for blunt end forming enzyme. Okay, so it make a cut in between A and T and it forms blunt end. Okay, then H3. Okay, so H3 is isolated from Haemophilus aegyptius. So this is its recognition sequence. It make a cut between C and G. So this is also straight cut. It form blunt end. Okay, hint 3 isolated from Haemophilus influenza 3. It's its recognition sequence. It make a cut in between A and A. This also make a sticky end. Then NOTI. So NOTI from Nocardia otidis caviarum, and this is its recognition site, and its cleavage site is in between C and G. Okay, so you should know the difference between recognition sequence and cleavage site. Okay, so the recognition sequence means it is a sequence which is recognized by the enzyme, and cleavage site. Okay, cleavage site is nothing but it is the cut, the location where the Restrictionn enzyme make a cut is known as cleavage site. Okay, then PST1 is isolated from Providenica stewarti. This is its recognition sequence. It also make sticky end. Then PVU2. This is isolated from Proteus vulgaris. So this is its recognition sequence, and it make a cut in between G and C, makes blunt end fragments. Then the last one is TDH31. Okay, so this is isolated from Thermus thermophilus. So this is its recognition sequence, and it make a cut in between 4th and 5th bases. Okay, so here it has 5-GAC, then N and N. Any one of the 4 bases it may have. Okay, but the cut is in between 4th and 5th bases from the 5' direction. Okay, so here also from the 5' direction it is between 4th and 5th bases. Okay, thank you for watching my videos. If you like my videos, please do like, share and subscribe. Thank you.